Welcome to Bio Bits and Bytes.
Great to be here.
Today, we're diving deep into some really fascinating new research, stuff that could genuinely change how we approach cancers like leukemia, even solid tumors.
Absolutely. It's pretty cutting edge.
Our mission, as always, is to take this complex science and, pull out the most important bits for you, make it clear, make it engaging.
Exactly. We'll try and cut through the jargon and get to the core ideas. We're looking at some totally novel strategies in cancer therapy today.
And guiding us through this is a fantastic scientist who really knows this field inside out and, crucially, knows how to explain it well. Welcome.
Thanks. Happy to be here and chat about this.
So our goal today, really, is to understand these new angles by looking at something, well, frankly, quite surprising. These RNA-binding proteins showing up on the surface of cancer cells.
Right, not where you'd expect them at all.
It feels like a totally fresh take. Now, as we dig in, we're definitely going to hit some scientific terms. Would you be okay just briefly explaining those as we go? Maybe give us a feel for why they matter in this context for our listeners.
Absolutely. No problem at all. It's vital. We're all on the same page. So yeah, I'll define things and explain their significance as they come up. Happy to do that.
Brilliant. Okay, let's jump right in then. So the headline finding seems to be these RNA-binding proteins, RBPs, which normally live inside the cell, right?
That's right. Typically, nucleus, cytoplasm, internal machinery.
But they've been found on the outside of cancer cells. And apparently they're not just randomly there, they're sort of hanging out with these other molecules called glycoRNAs.
Yes, glycosylated RNAs, or glycoRNAs.
In these organized little clusters called nanodomains. It sounds quite specific.
It is. Very specific.
This was definitely news to me. Can we start with the RBPs themselves? What do RNA-binding proteins usually do inside a cell, and why is finding them on the surface such a curveball?
Okay, sure. So RNA-binding proteins, RBPs, there's a huge variety of them, but fundamentally they bind to RNA molecules. Their main role is controlling, well, basically everything that happens to RNA.
Like what sort of things?
You know how RNA is made, how it gets processed, where it goes in the cell, how it's used to actually build proteins, and even how it gets broken down eventually. They're the managers of the cell's RNA world.
Okay, managers inside the cell.
Exactly. Mostly inside.
Yeah.
Nucleus, cytoplasm. So finding them on the cell surface.
Yeah.
That's weird because the surface is usually for communication, for sticking to things, interacting with the environment, not usually RNA management.
It's like finding the company accountant working the front security desk.
Huh. Yeah, something like that. It's just not their usual posting.
Okay, that makes sense. So if the RDPs are the misplaced accountants, what about these glycoRNAs they're found with? What are they and why might they be important here?
Right. GlycoRNAs, it's pretty literal. RNA molecules that have sugar chains attached. Glycosylated means having sugars added. We've known for ages that proteins get decorated with sugars.
Like glycoproteins.
Exactly. But finding sugars on RNA is a much more recent discovery. Their precise function right there, next to the RBPs on the surface, is still, being figured out.
But any ideas?
Well, the thinking is they might be involved in how the cancer cells are signaling or interacting with their surroundings, maybe talking to other cells, maybe even hiding from the immune system.
Like little sugar-coated messages?
Sort of.
Yeah.
And perhaps the RBPs are there to help read or present these messages. It's an active area of research, but their co-location is definitely intriguing.
And the nanodomains. You mentioned these organized clusters. Tell us about those. Why should we care about these tiny patches?
Okay, nanodomains. Think super, super tiny, highly organized neighborhoods on the cell surface. We're talking billions of a meter across. Yeah, really small. And the idea is that specific molecules gather in these domains to work together more efficiently. So having these surface RBPs and glycoRNAs clustered together suggests they function as a unit.
And for therapies?
For therapies, this is potentially great news. If these nanodomains are specific to cancer cells and contain key targets, we could design drugs to hit just those neighborhoods.
More precision, fewer side effects, hopefully?
That's the hope, aiming for a specific, concentrated target instead of a broadside attack on the whole cell. It could make treatments more effective and potentially safer.
Okay, this is setting the stage nicely. Now, the research really zooms in on one specific RBP, doesn't it? Nucleophosmin or NPM1?
That's right, NPM1 stood out.
It seems to be a major player on the surface, or CSNPM1 as they call it, across different tumors, but particularly in acute myeloid leukemia, AML.
Yes, the AML connection is very strong.
Apparently, it's on the main leukemia cells, the blasts, and even those really tough-to-kill leukemic stem cells. but not really on normal blood stem cells. That sounds significant.
It's incredibly significant.
First, though, what's NPM1's day job inside a healthy cell?
Well, NPM1 is a real multitasker inside the cell. It's involved in making ribosomes the protein factories.
Right.
It helps other proteins fold correctly, it regulates genes, it even helps repair DNA damage. It's usually busy inside the nucleus, the cell's control center.
So again, finding it on the surface.
Very unexpected. It's like the factory manager suddenly appearing on the loading dock, directing traffic.
And this specificity in AML on the cancer cells, especially those stem cells, but not the healthy ones, that feels like a huge therapeutic window, doesn't it? For you listening, this could mean a unique address just on the cancer cell.
Exactly. That tumor-specific expression makes CSNPM1 a really, really attractive target. If you can design something, like an antibody, that only recognizes CSNPM1.
You could potentially kill the leukemia cells while leaving the healthy blood-making cells alone.
Precisely. Which could mean fewer horrible side effects compared to traditional chemo. And what's also quite promising is that this CSNPM1 seems to be there regardless of the specific genetic mutations driving the AML in different patients.
Ah, so it's mutation agnostic, as the paper puts it.
Yes, which suggests a therapy targeting it could work for a broad range of AML patients, not just those with one specific mutation. That's a big deal.
That really is important.
Yeah.
Wider applicability. And you mentioned it's not just leukemia, they found it on solid tumors too.
That's correct. While AML was a major focus, the studies did detect CSNPM1 on cell lines from various solid tumors as well.
So the potential reach could be even wider than blood cancers.
It certainly hints at that possibility, yes. Still early days for some tumor types, but the initial signs are there.
Okay, so you have this promising target, CSNPM1. The logical next step is trying to hit it, right?
Exactly. Develop a weapon.
And that's where this monoclonal antibody, MAb2, comes into the picture. Can you break down monoclonal antibody for us? How does something like AB2 work? Maybe think about how our own immune system uses antibodies.
Sure. So your immune system naturally makes antibodies. These are proteins designed to recognize and latch onto specific things, usually invaders like bacteria or viruses.
Like a lock and key?
Kind of, yes. Very specific recognition. A monoclonal antibody is basically a lab-made antibody that's engineered to recognize just one specific target, one keyhole. In this case, the CSNPM1 protein on the cancer cell surface.
So it's like a highly specific guided missile?
That's a good analogy, yes. So the researchers developed MIB2 in mice specifically to bind to this CSNPM1, and then they tested it.
And how did the preclinical results sounded pretty strong?
They were very encouraging across multiple different mouse models of AML using mouse leukemia cells, human leukemia cells in mice, even leukemia cells taken directly from patients and grown in mice.
Those patient-derived models are really important, aren't they?
They are. They give you a better idea if it might work in actual humans. And in all these models, ME2 showed robust anti-leukemia activity. It reduced the leukemia burden, improved survival.
And crucially, what about side effects? That's always the worry.
Right. And this was maybe one of the most striking findings. In the healthy mice treated with MA2, even at effective doses, they didn't observe any significant toxicity.
Wow. That's huge.
It is. It suggests a potentially very good safety profile, which is obviously critical for any new cancer drug.
The report also mentioned it seemed particularly good at targeting the leukemia stem cells, the LSCs. Why is hitting those specific cells so vital in treating leukemia?
Ah, yes, the LSCs. These are like the root cells of the leukemia. They're a small population, often resistant to standard chemo, and they're thought to be the ones that can restart the disease after treatment, causing relapse.
So if you don't get the LSCs, leukemia can just grow back.
Yep.
Exactly. Targeting and eliminating LSCs is considered really crucial for achieving long-term cures. The fact that AB2 preferentially hit these LSCs in the mouse models is very promising. It suggests it might cut the disease off at the root.
Like pulling out the weed, not just trimming the leaves.
Yeah.
Okay. And going back to solid tumors, briefly, did the AB2 show activity there too?
It did, but with that important caveat of specificity. They tested it in mouse models of prostate and colorectal cancer, where the cancer cells did have high levels of CSNPM1 on their surface.
Right.
And it worked. It showed anti-tumor activity. But then they tested it in a melanoma model where the cells didn't have surface NPM1.
But it did nothing.
Correct. No effect.
Yeah.
Which really reinforces that AB2 is highly specific for its target, CSNPM1. It only works if the target address is present on the cancer cell.
Fascinating. A very precise tool. So knowing CSNPM1 is there and targetable is great, but the researchers also dug into what else is around it on the cell surface, right? Using something called proximity labeling. Yes, they wanted to understand the local environment, the molecular neighborhood around CSNPM1. Proximity labeling techniques let you sort of tag proteins that are very close physically to your protein of interest.
Like finding out who its immediate neighbors are.
Exactly. And the results were really interesting. They found, as you might expect, other proteins associated with the cell membrane, but they also found a lot of components from ribonucleoprotein complexes, or RNPs.
RNP, so more RNA-related proteins even out there.
Yes, and this was the striking part. In the AML cells, a huge chunk, something like 60 to 71% of the proteins found right next to CSMPM1 were other RNA-binding proteins.
60 to 70%. Wow.
Yeah, a really high proportion. And these neighboring RBPs were often the same ones found across different AML models.
So it really suggests CSMPM1 isn't just floating around randomly. It's embedded in these specific RBP-rich nanodomains you mentioned earlier.
That's the picture that emerges, yes. It seems to exist within a distinct community of RNA-related proteins right there on the leukemia cell surface. This might be important for its function, or perhaps for how accessible it is to therapies like GT2.
It really paints a picture of a complex, maybe vulnerable hotspot on the cancer cell, which leads us nicely into the broader role of RBPs in leukemia, not just the surface ones. The research emphasizes that alterations in RBPs generally are implicated in cancers.
Absolutely. Beyond this surface phenomenon, there's growing understanding that RDPs are critical internal players in driving leukemia and other cancers because they control so many steps in RNA processing and function.
Messing with them can really mess up the cell.
Precisely. If their activity is altered, if they're mutated or the levels change, it can have massive downstream effects, often contributing to uncontrolled cell growth. We see RBP alterations linked to many human diseases, and cancer is definitely a big one.
And there are different flavors of RBPs doing different jobs with RNA, aren't there? The research mentioned RNA modification and mRNA splicing. Can you unpack those a bit, maybe with an example or two relevant to AML?
Sure. So one major class involves RNA modification. These RBPs chemically change RNA molecules after they're made. A common modification is called M6A methylation.
M6A.
And you have RBPs that act as writers to add this M6A tag, erasers to remove it, and readers that recognize it and trigger some downstream effect.
Like editing marks on a script?
Kind of, yeah. These marks can affect RNA stability, translation, splicing, lots of things. For example, in AML, an M6A writer called MET-TL3 can boost the production of cancer-promoting proteins like CMYC and BCL2.
So tweaking the RNA messages to favor cancer growth. What about the other group, mRNA splicing RBPs? What's splicing and how do RBPs fit in?
Okay, mRNA splicing. This is fundamental. When a gene is first transcribed into RNA, that initial copy contains coding bits, exons, and non-coding bits, introns. Splicing is the process of cutting out the introns and stitching the exons together to make the final messenger RNA, the blueprint for a protein.
And RBPs do this stitching.
They are crucial components of the machinery, the spliceosome, that does this. Specific RBPs, called splicing factors, recognize the signals that mark the boundaries between introns and exons, guiding the cutting and pasting. Think of them as editors, ensuring the final message is correct.
And if the editors mess up?
If these splicing factors like SF3B1 or U2AF1, which are often mutated in AML, go wrong, you get missplicing. This can create abnormal proteins or fail to produce essential ones, contributing directly to cancer. They might mess up the splicing of key leukemia genes like HOXA9 or BMI1.
It sounds like incredibly precise work, easily disrupted with big consequences. The research also mentioned using CRISPR-Cas9 screens. That's the gene editing tool, right? How do they use it here?
Yes. CRISPR-Cas9 is like a pair of molecular scissors for DNA. Researchers used it to systematically knock out or disable individual RBP genes in AML cells one by one.
To see what happened.
Exactly. They look for which knockouts cause the AML cells to die. If disabling an RBP kills the cancer cell, it means that RBP is essential for the cancer's survival.
Finding the Achilles heels.
Pretty much. And this approach identified RBPs like RBM39 and DCPS as being critical dependencies in AML. This makes them really interesting druggable targets if you can inhibit them pharmacologically.
You might selectively kill the AML cells. Okay, what about the len 28 proteins they came up to? How do they fit into the cancer picture, especially with microRNAs?
len 28A and len 28B. These are RBPs known for their roles in stem cells and development, maintaining pluripotency. One key way they do this is by controlling the processing of a family of tiny regulatory RNAs called LET7 microRNAs.
MicroRNAs, those are the small RNAs that can silence other genes, right?
Exactly. They act like volume controls, turning down the expression of target genes. LIN28 basically blocks the production of mature, functional LET7 microRNAs.
And that's bad in cancer.
It can be, because LET7 normally keeps several cancer-promoting genes in check, like KRAS and CMYC. So if LIN28 is overly active, as it is in some leukemias and other tumors, you get less LET7 and consequently more of these growth-promoting proteins. LIN28 essentially takes the brakes off cancer cell growth.
Wow, it's such an intricate web of control. But it seems like researchers are actively trying to target these various RBPs. The paper mentions small molecule inhibitors.
Yes, and that's really encouraging. There's a growing pipeline of small molecules being developed or tested that target various points in this RNA regulation network. Inhibitors for M6A enzymes like MET-TL3, splicing factors like SF3B1, proteins like RBM39, even LIN28. It shows that the field views these RBPs as genuine therapeutic targets.
Which brings us to a specific strategy mentioned towards the end, targeting splicing factors, particularly RBM39, in AML cases that already have mutations in other splicing components. Can you explain that connection?
Right, this is quite neat. So mutations in core splicing machinery components like SF3B1 or U2AF1 are common in myeloid cancers like AML. We call this spliceosomal mutant AML.
So their splicing is already a bit wonky.
Exactly. Now, RBM39 is another RBP involved in splicing many RNAs, including ones important for AML survival. What studies found is that these spliceosomal mutant AML cells are often hypersensitive to drugs that cause RBM39 to be degraded or destroyed.
More sensitive than normal cells or other AML cells?
Yes, preferentially sensitive. It seems that because their splicing machinery is already compromised by the mutation, hitting them again by removing RBM39 is particularly lethal. There are drugs, certain sulfonamides, that work with another protein called DCAF15 to specifically trigger RBM39 degradation.
So it's like kicking a chair that's already missing a leg.
That's a good way to put it. You're exploiting a pre-existing vulnerability created by the splicing factor mutation. It's a great example of how understanding the specific molecular defects in a cancer can lead to tailored therapeutic strategies.
It really highlights the potential for personalized medicine based on these intricate RNA pathways.
Definitely.
Well, this has been an absolutely fantastic deep dive. We've covered so much ground, from the really surprising discovery of RBPs on the cancer cell surface
To identifying CSMPM1 as a very exciting potential target in AML and maybe other cancers.
and exploring the broader critical roles of various RBPs inside the cell, controlling RNA modifications, splicing, and more.
And that antibody, MA2, targeting CSMPM1 looks particularly promising, especially with its potential safety and ability to hit those leukemia stem cells.
Absolutely. The whole idea of targeting RNA regulation feels like a really dynamic new frontier in cancer therapy.
It really does. There's huge potential for novel drugs and strategies emerging from this area.
Which leaves us with a final thought for you listening. Given how incredibly complex all this RNA regulation is, how might targeting multiple RBPs or different RNA pathways at the same time potentially lead to even better cancer treatments down the line? And maybe what are the big challenges in actually developing those kinds of combination approaches?
That's the million-dollar question, isn't it? Combining targets could be much more powerful, perhaps preventing resistance. But the complexity is immense. Understanding how these pathways interact, predicting side effects, it's a huge challenge. It'll need really clever science and careful clinical testing to get there. But the potential payoff is enormous.
Something for us all to keep an eye on. Thank you so much for walking us through all of this today. Your explanations have been incredibly clear and helpful.
It was my pleasure. Really enjoyed the conversation.
And thank you all for tuning in to Bio Bits and Bytes.